This project is on the molecular genetics of tumor viruses by Baltimore, Delbecco, and Taman. Biologist David Baltimore, geneticist Howard Taman, and virologist Renato Delbecco shared the Nobel Prize for Physiology or Medicine in the year 1975 for their work on the molecular genetics of tumor viruses. The focus of this presentation will be on David Baltimore. As far as the knowledge prior to the 70s, they thought that DNA was the only medium for storing genetic information. Also, the Watson and Crick central dogma consisted of the idea that there was a one-directional flow of information from DNA to RNA and subsequently protein. It was discovered in 1971, however, that certain viruses are capable of shifting their genetic information from RNA to DNA. During the process of infection, such viruses, called retroviruses, transcribe the RNA genetic code back to DNA, followed by RNA and then protein. For the reverse transcription from RNA back to DNA, the retroviruses use an enzyme called reverse transcriptase, which is a specialized polymerase and constructs a double-stranded complementary DNA molecule from the RNA template. As far as the biography of David Baltimore, he was born in the year 1938 in New York City. As a child, he was both industrious and inquisitive, with a fascination for biology at the micro and the molecular level. His interests include watching golf and going fishing, as he does not spend every day in the lab. He's also very ambitious and assertive, as well as dynamic and capable, which can be demonstrated by his behavior during the controversial Imanishi-Kari case. In the controversial Imanishi-Kari case, Kari, an assistant in David Baltimore's lab, was accused of fabricating results for a published paper which described injecting a mouse with a gene, altering its immune system, thereby allowing it to produce antibodies against a given bacteria or virus. The accusation was made by an MIT postdoctoral researcher named O'Toole. Given his nature, his endangered reputation, and association with the case, Baltimore vehemently defended Imanishi-Kari. Although Kari was found guilty, the paper was re-examined in 1996, and she was vindicated, as it was found that the publication did not have any fraudulent data. As far as the education and accomplishments of Baltimore, during his high school years, he joined a research program at the Jackson Memorial Laboratory in Bar Harbor, Maine. This was his first experience working in a lab with scientists in the beginning of his research career. He also studied at Swarthmore College with a major in biology and then switched to chemistry. He became one of the first undergraduate research students at the Cold Spring Harbor Laboratory in 1959, and this is where he became introduced to the novel field of molecular biology. He also pursued his PhD in biophysics at MIT, and then he left MIT in 1961 to study animal viruses at Rockefeller University. He conducted graduate work there with Richard Franklin, who demonstrated how certain viruses can shut down cellular synthesis of RNA and then induce synthesis of viral RNA. Baltimore, as a postdoctorate, studied viral systems with a focus on viral RNA synthesis. He became a research associate at the Salk Institute in 1965, where he worked on the poliovirus. He discovered that the RNA of the poliovirus became the messenger RNA upon entering the cytoplasm of a host cell. He then became an associate professor of microbiology at MIT in 1968, and a full professor in 1972. And then he became the president of Rockefeller University in 1990. He was also the president of Caltech from 1998 to 2006, and then president of the American Association for Advancement of Science in 2007, and he's currently still a professor of biology at Caltech. As far as the research and discovery, during the 1970s, Baltimore collaborated on work with the Rous sarcoma virus, or RSV, as well as other tumor-causing viruses. RSV causes tumors in chickens. And so when an extract from a chicken tumor was filtered to remove anything larger than a virus, and the filtrate was then injected into lively chickens, tumors were produced. It was concluded from the study that virus was the cause of the tumors. However, they were uncertain of the mechanism. Timon incorrectly proposed that the tumor virus integrates its DNA into the host cell's DNA. However, RSV had its genetic information stored as RNA, so that posed a problem. David Baltimore was intrigued by how the RNA can be used as a template to synthesize DNA. The following procedure was conducted to investigate. So Baltimore removed the outer core of the virus and placed the inner core in a cell-free system. He then added all the nucleotides and used thymine as a radioactive tracer, since DNA contains the thymine base while RNA does not. The result was that radioactive DNA was produced by the viral components in the cell-free system, and it was confirmed that DNA was indeed produced because the product was unaffected by RNAs, but it was destroyed by DNAs. Baltimore also conducted further investigations. He incubated the viral cores with trypsin, which is an enzyme that digests protein. He then added nucleotides to the trypsin-digested viral cores and found that no DNA was produced. He also found that if he had added RNAs at the beginning of the experiment, no DNA was produced either. So based on his investigations, he concluded that RSV is a special enzyme which synthesizes DNA from an RNA template, and he called it the RNA-dependent DNA polymerase, but we now know it commonly as reverse transcriptase. Moreover, Crick's central dogma doesn't truly apply to retroviruses, as a reverse flow of information from RNA to DNA occurs prior to the one-directional flow from DNA to RNA to protein. Now, as far as the structure of retroviruses, they have an outer envelope, an inner capsid, and a central core that contains the RNA template and the reverse transcriptase enzyme. So here's a mechanism of how retroviruses cause infection. First, the retrovirus viral envelope fuses with the cell membrane of the host cell, and the capsid disassembles, thereby releasing RNA as well as the reverse transcriptase enzyme into the cytoplasm of the host cell. And then, the reverse transcriptase uses RNA as a template to synthesize a complementary DNA strand, after which it degrades the RNA and subsequently proceeds to synthesize a second DNA strand. So now the DNA goes into the nucleus of the host cell and integrates itself into the host cell chromosome. As far as the components and purpose of the retrovirus RNA, a typical retrovirus RNA has long terminal repeat sequences, or LTR, at both ends of the RNA, which help it to integrate into the host cell genome. After integration, the virus uses the host cell machinery to produce more of the viral RNA. The retrovirus also contains only 3 genes known as GAG, POL, and ENV. The POL gene codes for the reverse transcriptase enzyme, the GAG gene codes for the capsid and additional core proteins, and the ENV codes for outer envelope components. As a result, the proteins are assembled into a viral particle with the viral RNA inside. Once the virus leaves the cell, it can then affect other cells. Tumor viruses, like RSV, are unique because they have an additional 4th gene known as the oncogene, called SRC, and this contributes to malignancy. And some interesting facts are that retroviruses can be used as vectors in gene therapies. They can actually be engineered to carry genes into the body. And another fact is that HIV is actually a retrovirus which causes AIDS, and it has a considerably high mutation rate.